Hello everyone, welcome to my channel again. So in this video we will going to learn about the lymph field. Now lymph field is very important to understand the tetrapod limb development, because generally we ignore this part in CSIR as no questions have been asked from this area. But this is also important, and in future questions might be asked. So lymph field, as the name says, that this is a field which give rise to the limb. So this region in an embryo, there is a disc of cells, and it has all the cells, or it has the cells that are capable of giving rise to a limb. So where is the lymph field situated? So this is the lateral plate mesoderm. Okay, this is the lateral plate mesoderm region. Now in this region there is this disc of cells. What kind of cells are there? The group 1, which is the peribrachial flank tissue, this which is present on the left side. Then on the right side there is group 3, that is shoulder, that forms the shoulder girdle of the limb. But the limb itself forms from these central group of cells, that is the group 2, and this forms the free limb. But so all these three regions are forming limb and its accessory parts. But there is an additional ring of cells that surrounds this lymph field, and its role and its importance has been demonstrated from an experiment. Okay, so there was an experiment. First, researchers did what? They removed all these 1, 2, 3 group of cells. So whatever was inside, all these cells was removed, and they concluded that a limb will still form. So there will be a limb. There will still be a limb. How? Because of this additional ring of cells. Here, these additional ring of cells plays the important role. And these additional region, they give rise to a functional limb. They are capable of giving rise to a functional limb. But this will be a slower development. Okay, this limb will develop very slowly as compared to the normal scenario. Then what did they do? Like previous experiment, along with group 1, 2, 3, they also removed the group 4 cells, and they observed that there will be no limb development. Okay, so now when these additional, maybe these are the backup cells, now they are also been removed, so there will be no limb formation. So from this experiment, they have demonstrated that all of these cells are capable of giving rise to a limb, but normally all these cells which are inside the disc, they give rise to a functional limb. Now, so the limb bud from this lymph field, the limb bud will generate. Now, limb bud does not constitute only of lateral plate mesoderm. So it has This precursor cells are derived from three sources. What are those? First, the posterior lateral plate mesoderm. Second, some cells also come from the adjacent somites. And third, cells that overlie the bird's overlying ectoderm. Now, these mesodermal cells from the lateral plate mesoderm and from the somites, they form the mesenchymal cells, and they migrate from their respective locations. They migrate under the limb bud. So this is the limb bud, and they migrate under the limb bud. Here they proliferate. So the lateral plate mesoderm, they give rise to skeletal precursors of the limb, while the somite precursors, the somite cells, they give rise to muscle precursors. So here above the limb bud there grows the ectoderm. The ectodermal cells thicken makes a gap around the limb bud, and there is the proximal to distal axis. There is the proximal to distal axis which has been set. So in a limb bud, the first axis that is set is the proximal to distal axis. Proximal means from somite, and distal means to the ectoderm direction. So this is important. So there is more growth along proximal to distal axis, but there is less growth along dorsoventrally and anterior to posterior axis. So there is still growth. Initially there is still growth, but more growth is in the proximal to distal direction. Then the limb bud has three functional domains. What are those? One is the progress zone. The progress zone constitutes all these mesenchymal cells which are highly proliferative. They proliferate and they grow the limb bud itself. Then another zone is the JPA, or the zone of polarizing activity, and it is the most posterior of the progress zone, which forms the anterior and posterior axis of the limb, which we will going to deal in future. Another zone is the AER, or the apical ectodermal ridge, and this is the thickening of ectoderm at the apex of the limb bud. So these are the three functional domains, and the importance of the limb field we have studied here. So this was all about this part, and we will be going to learn about the different axis formation in future. Thank you for watching this video, and if you like this video, give it a thumbs up, or if you have any doubts, comment in the comment section below. Thank you for watching.